let's get into viruses and prions. So the purpose of this lecture is to help you understand what these things are and how they work. In Unit 4, we're going to look at individual examples of them and think of them as diseases. We'll look at the diseases they cause and things like that. But here, we're going to learn how they work and what makes them different from bacteria or anything else. And we've all heard of viruses, so I want to start with some big picture slides, and I want to make sure we are all on the same page about what a virus really is. So a virus is information. It's a nucleic acid sequence. So what are some nucleic acids? You should be able to name RNA and DNA. Those are the nucleic acids we think about most. And so a virus is just one of those sequences, and it encodes information that reprograms a cell. And it forces that cell to make virus particles, or what we call virions. So a virus really is the information that encodes controlling a cell, and it encodes how to make a virion. So it is just that information. We can, in a laboratory, make any virus. We can't design viruses. That's beyond what we understand how to do, but we could take a virus sequence, a nucleic acid sequence, and get it started. So again, I want to make that clear. As far as I know, no one on Earth knows enough to design a new virus. They're all way too complicated for any of us to understand. But we can take the nucleic acid sequence and put it in a cell and get it to start acting like a virus. You can't really do that with any other living thing. You can't take the DNA from a human cell and put it in a test tube and get a human cell to start being a cell. But viruses are simpler. Virions are the things we draw pictures of. If you've seen a picture of COVID with those spikes and the RNA inside it, that's a virion. So that's the form that moves between host cells. That's the form that takes the nucleic acid from one cell to the next. So I want to show you kind of an analogy I use. We can think of a dandelion plant. And you may be familiar with this. When a dandelion plant is done being a flower, it makes these seeds that are blown away on the wind. And a seed, its job is to carry the DNA and whatever else is necessary so that when it lands in a good place, a new plant will grow. And a virion, or virus particle, those mean the same thing, it's the same idea. Its job is to take the nucleic acids from one place to another place. And then the nucleic acids direct everything else. So if that was a virion, this is a virus. It's all the different possible forms, from that seed floating in the air, the seed when it is planted in the ground, to the whole plant with the roots and the leaves and the flower. Because the nucleic acids, the DNA in this case, carried in this seed, encodes the information for making all of this. So this whole thing would be analogous to the virus, while this would be the virion or virus particle. So this is one part of a virus's life cycle. So we do want to be careful in how we talk about it and what we're saying. And the virions, again, all they do is deliver nucleic acids to a new cell. And that's all they are capable of doing. The nucleic acids pretty much have to do everything once they get into a host cell. And so the complicated stuff that viruses do happens inside a host cell. And so to understand viruses, we have to understand host cells in a lot of detail. And we won't really do that in this course, and you may not you may not ever do that, but if you want to read the literature about viruses, if you want to read the articles coming out about how SARS-CoV-2 replicates, you need to know a lot about cell biology to understand that stuff. You need to understand how cells work at a very detailed level to understand how a virus reprograms a cell. So what information is on the nucleic acids from a virus? What is encoded on the RNA or DNA inside a virion? Well, generally, those nucleic acids encode proteins, and the host cell is going to make those proteins. And they could be regulatory proteins that change the way a host cell acts. They could be the structural proteins that make up a new virion. They could be special enzymes that the virus needs for making new nucleic acids. And I'll walk you through this later on. When you see specific examples of this, come back to these slides and this will make more sense. We can compare viruses, virions, this stuff, to living things, and you'll see that viruses are not really living. So, first of all, virions are not cells. We don't think of them as alive. The vast majority of them, we wouldn't ever think of them as alive. They don't divide. They don't do cell division. They don't grow bigger and then split into two new cells. That is not a thing viruses ever do. They don't take in sugar from the environment and convert it into ATP. They don't do respiration. They don't do fermentation. They don't make vitamins. They don't make their own proteins. They don't have ribosomes. So they can't make proteins on their own. They need a host cell to make the proteins. The molecules on the surface of a virion exist just to get to a new host cell and evade the immune system. So that's really all you're looking at on the outside of a virion. All those spike proteins, all they're trying to do is hide from the immune system and then attach to a host cell. But virions themselves, they are non-modal. They don't have flagella or cilia. They don't move. They just bounce around in the air or in liquid. And they don't secrete anything. They don't release toxins that poison us. Bacteria do that. Fungi do that. Protists do that. Virions don't do that. Viruses don't do that. And because of this, we can't use things like antibiotics on them. They don't have a viral ribosome we can inhibit. They don't have peptidoglycan we can inhibit. And they don't have much in common with each other. So we can't really get one drug that's going to work against all viruses. The things you'll see a virus doing to the host range from damaging the cells to killing the cells to changing the way the immune system works, to all the way to changing the host cell DNA. And this is something we see. There are a lot of viruses that cause mutations in host cells, and some viruses directly will cause cancers. Yeah, that is the thing that we'll talk about later. So, size-wise, we can compare virus particles, or virions, to bacteria, and then compare those to a human cell. And so the human cells that you'd learn about in A&P are much bigger than a bacterial cell, and a bacterial cell is much bigger than most virions. So this cell has to do tons of stuff. This cell has to make its own energy out of chemicals it gets from the environment, and it has to make proteins, it has to make nucleic acids, it has to encode everything and synthesize everything. It has to grow and get bigger and then split into two new cells. These things don't have to do any of that. All they have to do is get their nucleic acid into here or into here. So we're going to look a lot at virions and about the parts of them and what they do. So here are the parts. There's going to be a core, which we don't talk about much, but it's the place where the RNA or DNA is. Viruses can have RNA genomes. That's another weird thing about them that you will see. We will talk a lot about capsids. Capsids are the proteins that surround the nucleic acids and protect them. And they're made up of proteins called capsomeres. And they act like self-assembling structures. So capsomeres come together around the nucleic acid and form a repeating structure of a capsid. Some viruses have envelopes. Those are lipid bilayers that wrap around the virion. And so like SARS-CoV-2 that causes COVID-19, it has an envelope that wraps around it. And that envelope can have a lot of different functions, as we will see. And then most virions have spike proteins. These are proteins that stick out the side. And most of what these do is attach to a new host cell. So here are some capsid shapes that we see in nature. And these are all based on the capsomere proteins and how they assemble kind of randomly. And what we see, a lot of them are icosahedral. So the capsomeres make one of these regular structures. And sometimes we'll have an envelope around it, but the capsid itself would be this shape. Other times we have spiral or helical capsids made from capsomeres that arrange themselves into helices. So there are several different viruses that are classified this way. A lot of times I'll tell you, when I describe a viral disease, I'll tell you the shape of the capsid because that's a way we can kind of classify them. I'll tell you like these would be helical, these would be icosahedral, these would be complex because they have multiple shapes at the same time. These are viruses that infect bacteria. They're called bacteriophages. The rest of them are not bacteriophages. So smallpox affects humans, poliovirus is humans, herpes simplex is humans. This is a virus that infects plants. This virus can affect all mammals. These are viruses, these are virions. These are photographs of virions that infect archaea, and they're different shapes. These look like wine bottles or clubs or something, and that's a capsid of a different shape. We don't really understand how they get those shapes. We don't know much about them because it's hard to grow them, and it's not that important. Viral envelopes are important, and they're important because they have a big impact on how viruses behave and what they do to host cells. And so, like I mentioned, they are made of phospholipid bilayers. So they are very similar to a cell membrane, but they have a very different function from a cell membrane. Viruses aren't doing a lot of transporting of things in and out of the capsid. So let's think about it. First, we'll talk about what they are and how they're made. Then we'll talk about kind of what they do or their function. So typically, they are stolen from the host cell, and that means they have whatever glycolipids or proteins were embedded in the host cell membrane. So just imagine taking a chunk of the host cell membrane, of cell membrane from any one of your cells. It's going to have a lot of different proteins embedded in it. And so that's what a typical viral envelope is like. It just has whatever was in the membrane. And then the virus has also added spike proteins to it. So the spike proteins stick out and have important functions for attachment and penetration, as you will see. So they have several purposes that we will see a little better when we look at viral replication, but they are very important for that. One term you will hear for viruses that are not enveloped is naked. That just means viruses that never have an envelope. And it matters whether a virus is naked or has an envelope because that's really going to determine how it enters a host cell. And again, this will make more sense when we get to viral replication. And I'm going to tell you something now that I want you to file away for when we encounter it later. I'm going to tell you something about viruses that infect bacteria. Those viruses are called bacteriophage. And they have a different way of entering host cells than what animal viruses have. So animal viruses infect animal cells. So anything that infects us is an animal virus. But a bacteriophage will infect a bacterial cell. And there's a fundamentally different process there because our cells don't have cell walls, but bacterial cells do. And so viral replication kind of revolves around whether or not there's a cell wall. And you'll see all of this when we get to it. But the point I want to make here is that some bacteriophage are also enveloped and they don't have an obvious function. Those envelopes, it's not obvious how they get them and it's not obvious what their function is. But some bacteriophage do have envelopes. And as you'll see later on, bacteriophage don't enter cells the same way animal viruses do. So when you come back to this slide after learning about viral replication, the idea of an enveloped bacteriophage might seem very strange. It seems that way to me, but they do exist. So envelopes matter because they affect our ability to find the viruses using the immune system. They help the virus get into cells and they protect a virus. But there are two practical things to know about envelopes when you're thinking about protecting yourself from viruses. There's the idea of they are fragile. You can use ethanol to disrupt the envelope. And if it disrupts the envelope, the virus can't get into host cells. So that's why something like a hand sanitizer can be useful against viruses that have envelopes, because it disrupts the envelope. But those wouldn't work against naked viruses because naked viruses are surrounded by tough proteins that wouldn't be damaged that way. One example of a naked virus would be norovirus. This is a virus that spreads in outbreaks through things like dormitories and cruise ships and gives people severe vomiting. And something like hand sanitizer doesn't protect people from norovirus because the ethanol in hand sanitizers doesn't affect the capsid. It would only affect the envelope. And since norovirus doesn't have an envelope, there's nothing hand sanitizer is really going to do to norovirus. But something like SARS-CoV-2 or influenza, so the influenza virus, those are the viruses that cause COVID and influenza respectively. Both of those will be disrupted by hand sanitizer. As we get ready to take a break and then jump into the next video, I want to give you a sense of, again, what we're trying to accomplish and what we sort of need to think about before we can do that. So we're going to talk more about different aspects of virus biology that we would care about. And then we'll get into more what viruses do inside cells and how they do what they do. And the end goal there is for you to be able to describe how a virus works, what it does in a cell. And I want you to be able to explain to people how viruses are different from bacteria, how they are different from other cells. So that's one big thing I want you to get from these slides by the time we're done. So I will see you at the next video.